Okay, so beyond the amazing fact of self-replication, what else is totally essential for life as we know it? It's probably something you don't think about much, but honestly, without it, we wouldn't be here. The answer is kind of surprising. Efficient and really selective catalysis. Think about just, common table sugar. Sucrose. You have a bag of it, it can sit on your shelf for years. Totally stable. But then you eat that same sugar, and your body instantly starts breaking it down to CO2 and water,
Yeah.
right? Releasing energy. Energy you're using right now to think, move, even taste. That conversion, well, thermodynamically it wants to happen. It releases energy. But on its own, it's just incredibly slow. Way too slow to be useful. Your body can't wait decades for energy.
And that right there, that's the key, catalysis. Without these molecular catalysts, these vital reactions like breaking down sugar or, say, building new DNA, they just wouldn't happen fast enough for life, not even close. And this is where enzymes step in. They are genuinely remarkable, highly specialized proteins. They basically make almost every reaction in your cells possible, on demand, and incredibly fast.
Exactly. So that's our mission for this deep dive. We're going to unpack the amazing world of enzymes. We're pulling insights mostly from Chapter 6 of Lehninger Principles of Biochemistry, a real cornerstone text. We'll dig into how they work at a molecular level, these ingenious mechanisms they use, how we measure what they do, and crucially, how they're controlled inside you. Think of it like a shortcut, to understanding the molecules that orchestrate life itself. Okay, so let's start unpacking. The basic idea. Enzymes are powerful biological catalysts.
Powerful is almost an understatement. I mean, the rate accelerations are just staggering, often way beyond synthetic catalysts. We're talking like speeding reactions up by factors of 10 to the 17 sometimes. Imagine, okay, a reaction that normally takes maybe 78 million years to get halfway done. An enzyme can make that happen in milliseconds. It's really mind-blowing. But here's the absolute critical part. Like any catalyst, enzymes lower the energy barrier, the activation energy. They never change the overall reaction equilibrium. They don't change how much energy is released or consumed overall. They just get you to that equilibrium point, well, incredibly faster.
And they do it with amazing specificity. It's not just about speed, it's precision too. Each enzyme usually only works on one or maybe a few very similar molecules, highly specialized tools in a busy factory.
Exactly. And they have these special pockets, tailor-made pockets called active sites. That's where the substrate, the molecule being worked on, binds and gets converted into product.
And most of these enzymes are proteins, these complex folded chains of amino acids. Their function totally depends on that specific 3D shape. If you mess up the shape, like with heat, the enzyme stops working. Delicate machines.
That's right. And sometimes these protein machines need a little extra help to do their job. They might need simple inorganic ions, like magnesium, zinc, iron, we call those cofactors. Or they might need more complex organic molecules, coenzymes, which are often made from the vitamins in our diet. These coenzymes act like little shuttle buses, carrying chemical groups around during the reaction. When the enzyme protein part, the apoenzyme is joined up with its necessary cofactor or coenzyme. That complete active unit is the holoenzyme.
Okay, so it makes sense they needed a system to name and classify all these things. You often hear names ending in aces, right? Like urease breaking down urea. But there's also this formal international classification, the EC numbers, grouping them by reaction type, like transferases, hydrolases.
Yeah, 7 major classes now. Oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, and even translocases for moving things across membranes. It reflects the sheer diversity of jobs they do. Without this precise, categorized action, cellular processes just wouldn't happen at the speed life demands. It's fundamental.
All right, so let's go deeper. How do they actually work molecularly? We know uncatalyzed reactions are too slow.
Okay, picture a reaction like a journey over an energy hill. You start at the ground state, your reactants. To get to the products, you have to go over the peak. That peak represents the transition state. It's this fleeting, really unstable arrangement of atoms halfway between reactant and product, not something you can bottle. And the height of that hill, that's the activation energy, the barrier you have to overcome.
And the enzyme's core job is to lower that barrier.
Right.
It makes a lower path, an easier route over the hill, increases the rate. But crucially, it doesn't change the starting or ending energy levels, doesn't change the overall degrees. So the reaction equilibrium stays the same. It just gets there way, way faster, like digging a tunnel through the mountain.
Exactly like a tunnel. And the secret sauce is something called transition state complementarity. See, the old idea was like a lock and key. The enzyme perfectly fits the substrate. But think about that. If an enzyme hugged the starting substrate too perfectly, it would actually stabilize it, make it harder to react. Imagine our enzyme is trying to break a stick. If it binds the straight stick perfectly, it makes it harder to bend and snap. So the modern view is that the enzyme is most complementary, fits best with the transition state. That means the best, strongest interactions between the enzyme and the substrate happen only when the substrate is in that high-energy, bent, halfway broken state. It's like the enzyme preferentially binds to and stabilizes that difficult transition state, effectively lowering the energy needed to reach it.
And where does the energy for that stabilization come from? That's the binding energy, right?
Precisely. It's the free energy released when all those little non-covalent interactions form between the enzyme and the substrate. We're talking hydrogen bonds, ionic interactions, hydrophobic effects, lots of weak forces adding up. This binding energy is the currency the enzyme uses to pay for lowering the activation energy. And it doesn't take much. Lowering the activation energy by just about 5.7 kilojmol speeds up a reaction tenfold. A single weak interaction can provide anywhere from 4 to 30 kilojmol. So by forming multiple optimized weak interactions specifically with the transition state, enzymes can easily achieve those massive rate enhancements, lowering the barrier by 60, even a 100 kilojmol.
And that binding energy also explains the specificity we talked about. If the active site is perfectly shaped to bind the transition state of one specific molecule, it just won't bind the transition states of other differently shaped molecules nearly as well. Tailored glove, yeah.
Absolutely. And binding energy does even more. It helps reduce entropy, basically. It grabs the substrates and holds them in just the right orientation for a reaction. Less randomness, more productive collisions. It helps strip away the wire molecules surrounding the substrate desolvation because water can sometimes get in the way. It can even contribute energy to physically distort the substrate towards its reactive form. And then there's this really elegant idea, induced fit. The enzyme itself isn't rigid. When the substrate binds, the enzyme can change shape, clamp down almost. This brings all the catalytic groups into perfect alignment. Hexokinase, the glucose phosphorylating enzyme, is a classic case. It literally wraps around the glucose.
Okay, so beyond just binding energy and fit, what other chemical tricks do enzymes use? You mentioned acid-base stuff, covalent bonds.
Right. Many enzymes use general acid-base catalysis. They position specific amino acid side chains like histidine or aspartate perfectly in the active site. These act like proton donors or acceptors at just the right moment, stabilizing charged intermediates that form during the reaction. Very common. Then there's covalent catalysis. Here, the enzyme actually forms a temporary covalent bond with the substrate. This creates a whole new reaction pathway, one with a lower activation energy. And finally, metal ion catalysis. Super important. Maybe a third of all enzymes need metal ions. These ions can help hold the substrate in place, stabilize negative charges in the transition state, or even directly participate in redox reactions, transferring electrons. It's a whole toolkit.
Okay, shifting gears a bit, how do we actually study this? How do we measure enzyme activity? That takes us into kinetics.
Yeah, enzyme kinetics is crucial. It's about measuring reaction rates or velocities and seeing how they change. Typically, we measure the initial velocity, V0, right at the beginning before products build up much. And we look at how V0 changes as we vary the substrate concentration, S, while keeping the enzyme amount constant. Usually get this characteristic curve. The rate increases with S, but then it starts to level off. It approaches a maximum velocity, Vmax, when the enzyme is basically saturated, working as fast as it can.
And that relationship, V0 versus S, is described mathematically by the Michaelis-Menten equation, a cornerstone equation. It involves Vmax and another key constant, K-Lero, the Michaelis constant.
Right. K-Lero has a useful operational definition. It's the substrate concentration at which the reaction rate, V0, is exactly half of Vmax. It can give you a sense of how much substrate is needed to get the enzyme working near its maximum rate. K-Lero varies vary wildly between enzymes, and while it's related to substrate affinity, it's not always a simple measure of it. It depends on the relative rates of binding, dissociation, and the actual catalytic step.
So how do researchers actually find Vmax and Km from their experiments?
Well, historically, they used linear plots, like the Lineweaver-Burk plot, which rearranges the Michaelis-Menten equation into a straight line. Makes it easy to see. Nowadays, though, with computers, it's more common and accurate to use nonlinear regression to fit the curve directly. From Vmax and the total enzyme concentration key, we can calculate another important parameter, kcat.
Kcat, that's the turnover number.
Exactly. It's Vmax divided by Kis. It tells you the maximum number of substrate molecules a single enzyme molecule can convert to product per unit time, usually per second, when it's fully saturated. Some kcat values are just incredible. Like catalase breaking down hydrogen peroxide, its kcat is around 40 million per second.
Wow. Okay, so if kcat is max speed and Km is about substrate concentration, how do you compare overall efficiency?
The best measure for that is the ratio Kcat-gamma. This is often called the specificity constant. It reflects how well the enzyme works when substrate is scarce, which is often the case in cells. Some enzymes are so efficient, their Kcat-gamma value approaches the physical limit of how fast molecules can diffuse together in water, the diffusion-controlled limit, around 108 to 109 M1S1. We call these catalytically perfect enzymes.
What about reactions with more than one substrate, which is pretty common, right?
Very common. These are bi-substrate reactions. They can happen in different ways. Sometimes, both substrates bind to the enzyme together, forming a ternary complex before any reaction happens. This can be ordered, where one has to bind first, or random. Other times, it's a ping-pong mechanism. The first substrate binds, reacts, and one product leaves before the second substrate even binds. The enzyme gets temporarily modified in between. You can actually tell these mechanisms apart by analyzing the kinetic patterns, how the rates change when you vary both substrate concentrations.
And things like pH matter too, don't they? You often see enzymes having an optimum pH.
Absolutely. Enzyme activity is usually very sensitive to pH. There's typically a narrow range where the enzyme works best. That's because the ionization states of the amino acid residues in the active site, those acids and bases doing the catalysis, are critical. Change the pH, you change their charge, and that can mess up the whole mechanism. And sometimes, to get really detailed info, scientists use pre-steady state kinetics. Looking at the very, very first moments of the reaction, like the first few milliseconds, you might see an initial rapid burst of product before the reaction settles into its steady rate. This burst can tell you which step is the slowest, the rate-limiting step, like maybe product release is slow.
Okay, let's pivot to inhibition. This is so important, especially for medicine and drug design. How do inhibitors work?
Well, broadly, there are reversible and irreversible inhibitors. Reversible inhibitors bind, well, reversibly. They can come on and off. The most common is competitive inhibition. Here, the inhibitor molecule looks structurally similar to the actual substrate and competes for the same active site. It increases the apparent kinoma. You need more substrate to reach half-max velocity, but it doesn't change the actual Vmax if you add enough substrate to outcompete it. The methanol poisoning example is perfect. Ethanol competes with methanol for the active site of alcohol dehydrogenase, slowing down the production of toxic formaldehyde.
Okay, so competitive is like blocking the parking spot. What are other reversible types?
There's uncompetitive inhibition. This is different. The inhibitor only binds to the enzyme substrate ES complex, not the free enzyme. It binds at a separate site. This effectively lowers both the apparent Vmax and the apparent kinema. And then there's mixed inhibition, where the inhibitor can bond to either the free enzyme or the ES complex, usually at a site separate from the active site. It affects both Vmax and kinoma, but in complex ways.
And the other major category is irreversible inhibition. Sounds more permanent.
It usually is. These inhibitors bind very tightly, often forming a covalent bond with a crucial amino acid in the active site. They essentially kill that enzyme molecule. A really clever type is suicide inactivators, or mechanism-based inactivators. These molecules aren't reactive on their own, but when the target enzyme binds them and tries to catalyze a reaction with them, the enzyme's own mechanism converts the inhibitor into a highly reactive form that then attacks and permanently inactivates the enzyme. The drug DFMO for African sleeping sickness is a great example. The parasite's enzyme basically commits suicide trying to process the drug. Another incredibly important concept, especially for drug design, is transition state analogs. Remember how enzymes bind the transition state super tightly? Well, these are stable molecules designed to mimic that unstable transition state. Because they resemble the state the enzyme binds best, they bind much more tightly than the actual substrate, often thousands or millions of times tighter. They act as potent inhibitors. This idea is fundamental to designing many modern drugs, like the HIV protease inhibitors we'll probably talk about.
Perfect transition. Let's look at some real-world examples. How does understanding these mechanisms translate into things like life-saving drugs? Start with chymotrypsin.
Right, chymotrypsin. It's a classic digestive enzyme, a serine protease, meaning it uses a key serine residue in its active site to cut peptide bonds in proteins. It employs that catalytic triad, serine, histidine, aspartate, working together beautifully. The reaction happens in two main steps: acylation, where the enzyme gets temporarily attached to part of the substrate, and deacylation, where water comes in to release the product and regenerate the enzyme. And it uses something called the oxyanion hole, specific features in the active site that stabilize the negative charge that forms on an oxygen atom during the transition state. Makes the reaction easier.
Okay, so understanding that detailed mechanism, how did that lead to something like HIV drugs? That seems like a big leap.
It's all connected. HIV needs its own protease, an aspartyl protease, to cut up its newly made viral proteins so the virus can assemble properly. Scientists figured out the structure and mechanism of HIV protease, how it uses two aspartate residues in its active site. Armed with that knowledge and the principle of transition state analogs, they designed molecules that look like the transition state of the peptide bond being cleaved by HIV protease. These molecules wedge themselves into the active site and bind incredibly tightly, stopping the protease from working. These HIV protease inhibitors were revolutionary. They block viral maturation and have turned HIV/AIDS from a near-certain death sentence into a manageable condition for millions. It's a triumph of rational drug design based on understanding enzyme mechanisms. And let's revisit hexokinase, the glucose enzyme. That induced fit is key. Remember, it needs to phosphorylate glucose, but not water, which is everywhere. When glucose binds, the enzyme undergoes this big conformational change, closing around the glucose. This brings the catalytic groups into position and excludes water from the active site. No glucose, no conformational change, no accidental phosphorylation of water. It's elegant discrimination.
And enolase, another enzyme from glycolysis, highlights the role of metal ions.
Yes, enolase absolutely needs magnesium ions, Mg2+. The Mg2+ ions do several things. They help position the substrate correctly, they make a proton easier to remove by stabilizing the resulting negative charge nearby, and they shield other negative charges during the reaction. They're acting as essential catalytic helpers, stabilizing that tricky transition state.
Okay, another huge medical area, antibiotics. How does penicillin work its magic?
Penicillin is amazing. It's basically an irreversible inhibitor of a bacterial enzyme called transpeptidase. This enzyme is crucial for bacteria because it cross-links the peptides in their cell walls, making them strong. Penicillin resembles the natural substrate of the transpeptidase. It enters the active site and the enzyme starts to react with it, but instead of finishing the reaction, penicillin forms a permanent covalent bond with the key serine residue in the active site. So the enzyme is dead. No cross-linking weak cell wall, and the bacterium often just lyases or bursts. Of course, bacteria fought back, evolving beta-lactamoses, enzymes that specifically chew up penicillin and related antibiotics before they can reach the transpeptidase. So what did we do? We developed other drugs, like clavulanic acid, which are themselves suicide inactivators specifically targeted at the beta-lactamases. Clavulanic acid gets processed by the beta-lactamase, turns into a reactive species, and kills the resistance enzyme. It's often given with penicillin-like antibiotic to protect them. It really is a chemical arms race.
Wow. Okay, so we have these incredibly powerful specific catalysts, but they can't just be running wild all the time. How is their activity regulated?
Regulation is just as critical as catalysis itself. Cells need to control when and how much enzymatic activity happens. Enzymes whose activity is regulated are often called regulatory enzymes. One major way is through allosteric regulation. This involves molecules called allosteric modulators, binding to the enzyme at a site other than the active site, the allosteric site. This binding causes a shape change that either increases positive modulator or decreases negative modulator, the enzyme's activity. If the modulator is the enzyme's own substrate, it's called homotropic regulation, often leading to those S-shaped kinetic curves, indicating cooperativity. If the modulator is a different molecule, it's heterotropic. The classic example is aspartate transcarbamoylase, ATCase. It's involved in making pyrimidines, building blocks for DNARNA. Its activity is inhibited by CTP, an end product of the pathway. That's feedback inhibition. When you have enough product, you shut down the assembly line. Sounds right. Conversely, ATP, which signals high energy and a need for nucleotides, acts as a positive modulator, speeding ATCase up. These modulators shift the enzyme between a less active T state and a more active R state.
So that's binding small molecules. What about actually modifying the enzyme itself?
Yes, reversible covalent modification is another huge regulatory strategy. There are hundreds of types, but the most common by far is phosphorylation. Specific enzymes called coking kinases attach a phosphate group, usually from ATP, onto serine, threonine, or tyrosine residues of the target enzyme. This adds a bulky, negatively charged group, which usually causes a significant conformational change. Altering activity could be activation or inhibition, depending on the enzyme. Then other enzymes called protein phosphatases remove the phosphate group, reversing the effect. It's like a molecular on-off switch. Think about glycogen phosphorylase, which breaks down stored glycogen. It's activated by phosphorylation when your muscles need quick energy. Multiple phosphorylation sites on some proteins allow for really fine-tuned integrated control.
And sometimes activation is, well, permanent, like cutting something.
Exactly. Proteolytic cleavage. Many enzymes, especially digestive enzymes like chymotrypsin or those involved in blood clotting, are synthesized as inactive precursors called zymogens. They're kept inactive until needed. Then, a specific protease comes along and makes one or more precise cuts, cleaving off a small part of the zymogen. This causes a conformational change that opens up or forms the active site, activating the enzyme. It's an irreversible activation step, useful for processes that need to happen quickly and decisively once triggered.
Which sounds a lot like the blood coagulation cascade. That seems like the ultimate example of regulation.
It really is. It's a fantastic example of a regulatory cascade where one activated enzyme activates many molecules of the next enzyme, which activates many molecules of the next, leading to huge signal amplification. A tiny trigger, like damage to a blood vessel, initiates a cascade involving numerous zymogens, clotting factors, being sequentially activated by proteolytic cleavage. There are intrinsic and extrinsic pathways that converge, all leading ultimately to the formation of fibrin, the protein mesh that forms the clot. The complexity is astounding, with built-in controls and inhibitors like antithrombin to prevent it from running wild and causing unwanted clots. Thrombosis. Drugs like warfarin, a vitamin K antagonist needed for some factors, or heparin, boosts antithrombin, target this cascade. And tragically, defects in the cascade, like missing factors, lead to bleeding disorders like hemophilia. So, connecting it all back, you see, the control of catalysis is just as fundamental to life as the catalysis itself. It's this regulation allosteric effects, phosphorylation, zymogen activation cascades that allows for the incredibly complex dynamic responsive system that we call a living cell or a living organism.
What an incredible journey. From just, basic enzyme principles, speeding things up, to how we measure that, the kinetics,
Yeah.
all the different chemical strategies they use, and then these layers upon layers of intricate regulation. It really is mind-boggling organization happening inside us right now.
Absolutely. And think about this. This detailed understanding allows us to design drugs, understand diseases, maybe even engineer new enzymes. What other complex biological processes rely on similar principles of regulated molecular interactions? And what future therapies might emerge from digging even deeper into these mechanisms? There's still so much to learn.
Yeah. So what does this mean for you listening? It means even the simplest things your body does.
Right.
From using that sugar you ate to just thinking. It's all built on this foundation of absolutely astounding molecular machinery, constantly working, constantly being tweaked and fine-tuned. Well, that wraps up another deep dive. We really hope exploring the world of enzymes has given you some valuable insights to chew on. Thanks as always for being part of the Deep Dive family. Until next time, keep digging deeper.